So we are starting with one of the most important tissue that is called as meristematic tissue. So whenever we talk about meristematic tissues, let's first learn the characteristics. So what I will be doing, I will be putting all the characteristics in flowchart form. So in the center, we are going to write characteristics. So we are going to study all the characteristics of meristematic tissues. Very first point that I can say that all the cells of meristematic tissues, they are living. Meristematic tissues, they have power of division. It means they can divide and they keep on dividing throughout their life. All the cells of meristematic tissues, they will have a dense protoplasm. They have a dense protoplasm. Then the cells of the meristematic tissues, I can say they have a prominent nucleus present. The cells, when we talk about the cells, so we can say that the cells are small in size. They all are isodiametric. Isodiametric means they have same diameter from all sides. Sometimes they are oval, or even I can say they are polygonal in shape. When we talk about the cell wall, so remember the cell wall of meristematic tissues, they are thin, they are elastic in nature, and they all are made up of cellulose. When we talk about the cells, how the cells are arranged, so I can say that all the cells of meristematic tissues, they are compactly arranged. When I use this word compactly arranged, means they are very close to each other, and most important that there is no intercellular space. It means between the two cells you are not going to see any space present. And what we can say in with respect to vacuoles, I can say very few vacuoles present, very few, very small, very few vacuole, or even I can say the vacuoles are absent. And the last characteristic, but the most important one, let me put it in another color, that the meristematic tissues, they have very high rate of respiration. So I can say that the rate of respiration is very high. So these all becomes the characteristics of meristematic tissues. Now if I have to study all these characteristics in the form of diagram, so I can put it in the diagrammatic form so that it becomes easy for every one of us to understand. So let me draw a diagram for meristematic tissues. So what I will be doing, let's take it in this way. Diagram for meristematic tissues. So first we will be drawing all these cells, most important. And you need to remember that these cells do not have any intracellular space. So I can say in between the cells you will not find any space. So this all space will be actually filled up by some. So these are all cells, and the cells do not have any intercellular space. There is no intercellular space present in case of meristematic tissues. So these are all the, you can say, meristematic tissues cells. And in this we are going to draw the cell membrane. So the black color one is the cell wall, which is very thin, it is elastic in nature, and it is made up of what? So I can say it is made up of cellulose. And this pink color one is cell membrane. The black color one, which is cell wall, remember it is freely permeable, it allows everything to enter. And the pink color one, the cell membrane, is selectively permeable. It means it will allow only selected substances to enter. Inside each cell there will be a prominent nucleus. So this nucleus is prominent. Prominent means that it is easily visible. You can see it very normally. And finally what we have is the protoplasm. So this is all dense protoplasm. Vacuoles in the meristematic tissue cells, it may or may not be present. If it is present, remember it will be very small. And most important, they do not have any intracellular space. So let us do the labeling now of meristematic tissues. So very first thing, I can say that all the cells of meristematic tissues, they are going to have thin cell wall, and their cell wall is made up of cellulose. After that, we can say that the cells, these cells that you can see here, the cells are small in size. They will be isodiametric. This is again one of the options. The cells are small, isodiametric, or even I can say they are polygonal. So this becomes with respect to the cell. There is no vacuole present here. Most interesting, there is no vacuole. And I can say they have a dense protoplasm. And inside the dense protoplasm, you are seeing a prominent nucleus. So there is a prominent nucleus present. And above this, there is no intercellular space. As you can see in the diagram, there is no intercellular space. So this all becomes the explanation for meristematic tissues. Now there are different types of meristematic tissues based on their position, based on their location, based on their origin. So let's look at the meristematic tissues based on origin. So very first, we are going to talk about origin. There are meristematic tissues based on origin. Origin means from where they have arisen. So based on origin, the very first one that we have is called as promeristem. This is the first one. Pro basically means first, so it is the first meristem. So when we talk about promeristem, I can say that it originates from embryos. The promeristem, I can say it originates from embryos. These promeristem, where they are found, you can say at the tip of stem and root. So small area basically at the tip of stem and root. So on the tip of stem and root, the meristematic tissue that you get is called as promeristem. This promeristem will further develop and it will differentiate and it will form what? I can say primary meristem. It means that the promeristem develops and it forms what? Primary meristem. So the moment we talk about primary meristem, very first point I will say that this primary meristem, it originates from promeristem. Remember, primary meristem, they all originates from promeristem. Most important in primary meristem that the cells are always active. When I say always active, it means that they are continuously dividing. And most important, that promeristem exists in plant from beginning. So right from the beginning and till the death of the plant, promeristem will be present. Where it is present, so we can say it is present at the tip of root and stem. So at the tip of root and stem, what you're going to get? You're going to get this. So remember this. This becomes the most important part here. It is present at the tip of the root and stem. It exists from the beginning. This is primary meristem. After primary meristem, we have the third one that is called as secondary meristem. So this is the third one called as secondary meristem. So logically, what do you mean by this and where they are located? That is what we need to remember here. So when we talk about secondary meristem, the primary meristem will develop into secondary meristem. So secondary meristem, it forms when it is formed. So we can say formed later during growth. So when the plants are growing, in the later stage, you will get secondary meristem. Even I can say that the cambium is seen. Cambium in both root and stem. So cambium in both root and stem will get secondary meristem. What is the role of the secondary meristem? I can say secondary meristem always increases thickness of plant. So if the plant is thick, it is because of secondary meristem. What is cambium basically? So if I take a tree and I cut this tree, so after cutting the tree, I see some rings present here like this. So the middle part basically here, the center one, center tissue that you can see, it is called as cambium. And this cambium is formed because of secondary meristem. So these are the meristematic tissues based on origin. Now we need to talk about meristematic tissues based on position. So let's talk about meristematic tissues based on position. So with respect to position, we need to draw a diagram that will help us to understand. So I draw this as the a branch, or I can say stem, and this just a cut to show some branching. So let us take this now. This is the branch which is arising. From here also we can show a branch. These are all branches. So some will be present at the tip here. This is also a tissue, meristematic one. Some will be formed at the node. So in this stem, you will see all nodes and internodes. So here what you can see, these are nothing but the nodes. And the space that you observe between two nodes, they are called as internodes. So here I can say these are nothing but the nodes. And the one that is responsible for increasing the girth. Girth means thickness of the tree. If you want the thickness to become tree to become fat, so with respect to girth, there are different tissue. So very first, based on position, the one which is present at the tip, it is called as apical meristem. So what are the things that we need to write? So first I will say that this apical meristem is nothing but, it is the growing point apex of secondary root and shoot. So I can say it is growing point apical meristem, and this apical meristem is nothing but, it is the apex or tip of secondary root and shoot system. You will get this apical meristem. Then what is the role? So we can say it divides continuously. The cells of this apical meristem divides continuously to increase shoot and root. So I can say that the length of shoot and root will be increased because of apical meristem. What is present in this apical meristem? So I can say it includes promeristem, very important. It includes promeristem and primary meristem. So remember, it includes promeristem and primary meristem. This becomes the apical. Now the next one is the one that is present between the nodes. So this, what you can see, is the second one. It is called as intercalary meristem. So it is called as intercalary meristem. Intercalary means, again, it's a part of apical meristem only. So very first point I can say for intercalary meristem, it is part of apical meristem. This is the first one. Second, I can say it gets separated when there is a permanent tissue formation. So I can say this intercalary meristem, it gets separated due to permanent tissue. So remember, it gets separated due to permanent tissue. And this intercalary meristem helps in growth of nodes and internodes. So the nodes and internodes, if they are growing, it is possible because of the intercalary meristem. The third type of meristem is this one, which we call it as lateral meristem. So the lateral meristem, as the name suggests, it is responsible for the lateral growth of the plant. So I can say this lateral meristem is responsible for, it is parallel to the sides of organ. So it is parallel to the side of organ. What is the other role of this lateral meristem? So I can say it increases the diameter of plant, very important. It increases the diameter of plant. And what it forms, it results in the formation of secondary permanent tissue. So lateral meristem results in formation of secondary permanent tissue. Means the cells that will not divide further, that becomes the permanent tissue. And at the last I can say that this lateral meristem is responsible for formation of vascular cambium, the cambium present between the xylem and phloem, and cork cambium, the one that is responsible for the girth. So this all becomes the meristematic tissues based on the position, where we have three types: the apical meristem, this is the first one. The second one is the intercalary meristem, that is present on the nodes. And the third one is the lateral meristem, which is responsible for increasing the girth of the plant. So this all becomes the meristematic tissues. Let's talk about the third type of meristematic tissues. It is based on function. Based on function. So there are different functions allotted to meristematic tissues, and based on functions we have different types of meristematic tissue. Very first type is called as protoderm, the one that is responsible for formation of outermost layer. Proto means first and derm means layer. So protoderm is nothing but, it is the outermost layer which is responsible for giving rise to epidermis. So remember, protoderm gives rise to epidermis. So epidermis arises only because of protoderm. Next we have procambium. So procambium means the initial one, the first one that is there. So cells of young growing region. Procambium is found at the cells of young growing region. So this young growing region is nothing but procambium. It is responsible for primary vascular tissue. So the initial xylem and phloem that is formed, it is formed because of procambium. And this formation takes place by two different methods. One is elongation, another one is differentiation. So by elongation and differentiation, there is a formation of procambium. The third is the ground meristem. Ground meristem means if I take any stem, so how the cells of the stem are arranged, means from epidermis to all the parts till the center, how they are arranged, that becomes your ground meristem. So ground meristem, it can be seen in young region, and this ground meristem is responsible for, so we can say that this ground meristem, it gives rise to, so the parts that ground meristem gives rise to, very first will be hypodermis. Inner to hypodermis you will see all cortex. Inner to cortex there will be endodermis. Inner to endodermis you will get pericycle. Inner to pericycle you are going to get medullary rays, and finally you will get pith. So this all becomes the ground meristem, and this is what entire meristematic tissues is all about. We are talking about next important one, that is simple permanent tissue. So in this simple permanent tissue, the very first one is called as parenchyma. So when I use this word parenchyma, the word para means beside. And when I use this word enchyma, enchyma means to pour. So this is parenchyma. Pairing on the side or beside we are pouring something. So let's look at the characteristics first. So in the center I will write parenchyma and let's highlight it. And now we are going to talk about the characteristics of parenchyma in detail. So very first, the parenchyma, I can say it is simple, common, unspecialized tissue. It is simple, it is common, and it is unspecialized tissue. When I use this word unspecialized means there is no specific function allotted to it. But this parenchyma is called as fundamental tissue. Why it is called as fundamental tissue? Because it is found in each and every part of the plant. Wherever you go, you will find parenchyma. Even the pulp of the fruit that you eat is parenchyma. The parenchyma tissues, I can say they are living. They have a prominent nucleus. Nucleus is easily visible and they have a large vacuole. So in meristematic tissues, vacuoles were absent, but in parenchyma, vacuoles are present. When we talk about the cells of parenchyma, they can be oval, they can be round, they can be polygonal, even they can be elongated, they can be star or irregular shape. So there is no fixed shape to the parenchyma cells. Depending on the nature, they will change. When we talk about cell wall, so remember in parenchyma, the cell wall is made up of cellulose and pectin. So I can call it as pectocellulose. And most important, between the cells, what you're going to get? Intercellular space. So in meristematic tissues, this intercellular space was absent, but here you will see intracellular space. And at the last, what we can say, that all cells are attached. So I can say all cells are attached to each other by plasmodesmata. So plasmodesmata is the most important component that will attach all the cells together. So let us draw the diagram of this parenchyma tissue. That will be easy for us to understand. So here what we are going to do, we are going to draw these cells. So these are all the cells of parenchyma tissue. We can draw now. See, there will be intracellular space present. So here we can show some intracellular space. So this is all intracellular space present. So this is all intracellular space. Now they have a cell wall. So inner to the cell wall, what we are going to get? So I will say inner to the cell wall there is cell membrane present. What is cell wall made up of? So we saw that the cell wall of parenchyma is made up of cellulose and at the same time it is made up of pectin. So pectin and cellulose both are present in the cell wall and that's why we say it is pectocellulose. The nucleus in case of parenchyma is very prominent and it is visible. And most important that these parenchyma cells or the cells of the parenchyma tissue, they show the presence of vacuoles. So this becomes the nucleus, which is a prominent nucleus seen in case of parenchyma. So this is the nucleus of the parenchyma tissue. And inside this you are going to get a, so I can say there is a vacuole present. So let us draw a vacuole. So the big one that you can see, it is nothing but the vacuole present. And all the plant cells of parenchyma, they are attached to each other. So they are attached to each other by the help of plasmodesmata. So this green color structure that you can see here is called as plasmodesmata. Plasmodesmata is like a door which is connecting the two cells. So both the cells, if they are connected to each other, They are connected to each other by the help of plasmodesmata, which is very prominently seen in case of parenchyma. And finally, we draw the cytoplasm. So here what you can see, this is nothing but the cytoplasm. So this is parenchyma tissue. Parenchyma cells don't have any fixed shape. They can be irregular shape, they can be oval shape, but they show vacuoles present. And in between the cells, you will see what intercellular space. So this is all what you should know about the diagram. Let us do the labeling so that it is easily visible. So very first what I can say, that the cells, they have a cell wall. Their cell wall is made up of two components. They are made up of cellulose or they are made up of pectin. Inside this cell wall, what you have cell membrane. Inside the cell membrane, there is a protoplasm. Now, when we talk about the protoplasm consists of a very prominent nucleus, and at the same time, they are going to have a very prominent vacuole. And all the cells are attached to each other by the help of plasmodesmata. So this becomes the diagram for the parenchyma. So this is parenchyma tissue. When we talk about the parenchyma, they are of three types. So I put it in this way. The parenchyma is divided into three types depending on the nature and position. So let's check it in this. This is the first one, second one, and the third one. Very first one I can say that it is Sclerenchyma. The parenchyma. The second one is called as Arenchyma. And the third one is called as Prosenchyma. So what exactly you mean by this? When I say Sclerenchyma, the word chlorine means green. So when the parenchyma tissue or the cells of parenchyma has a chloroplast, it is called as Sclerenchyma. And they are responsible in doing photosynthesis. So if the parenchyma has chloroplast, it is called as Sclerenchyma and will do photosynthesis. Arenchyma, as the name suggests, the word is air. So we can say there are large intercellular space. And this large intercellular space, basically, they all are filled with air. They all are filled with air. And because of the aeration, what happens? Buoyancy is offered. It means they can float on the surface of water. So basically I can say because of this aeration and buoyancy, the plants are able to float. So the all aquatic plants, they can float on the surface of water because of this Arenchyma. The next one that we have is the Prosenchyma. What do you mean by this Prosenchyma? So this parenchyma has a thick cell wall. And why it is thick cell wall? It is due to cellulose. There is more cellulose, no pectin as such. And what is the role of this Prosenchyma? So the duty of Prosenchyma is to provide strength and rigidity. So if there is strength and rigidity to parenchyma, it is because of Prosenchyma. Let's understand the different functions of parenchyma. So whenever we talk about the functions of parenchyma, very first function that I can say that it is found in basically root, stem and leaves. They are found in root, they are found in stem and they are found in leaves basically. Second, I can say it is responsible for forming the cortex of root, ground tissue in stem, then it is responsible for formation of pulp of fruit. Like mango, you eat the yellow part, so the yellow part that you eat, the pulp of the fruit, is nothing but the parenchyma. Third one, I can say that this is responsible for formation of pith. So I can say it is found or it is present in pith, the central space, and it is also acting as a packing material in xylem and phloem. So in xylem and phloem, if you see a packing material, it is because of the parenchyma. So this is all what you should know about the parenchyma. After this, we have the next important one, that is called as collenchyma. So let's talk about collenchyma. What do you mean by this word? When I use this word cola, the word cola means blue. Then we have the word enchyma. Enchyma means to pour. Again, the collenchyma is a living tissue. Collenchyma is a living tissue, but most important function is what you know. It is flexible and it is responsible for providing mechanical strength. If there is a strong wind blowing, the leaf will move with the tree. The leaf will flatter, but it will never fall down. It is because it is attached by the help of collenchyma. So if I take this part here and this leaf, so no matter how much is the wind, the leaf will not fall. Why it will not fall? Because they are attached to the stem by the help of this important tissue collenchyma. So let's look at the characteristics. So here in the center we will write collenchyma. So what are the basic characteristics that we need to study about collenchyma? First, I will say that the collenchyma is living, simple and permanent tissue. Collenchyma is living, it is simple, and it is a permanent tissue. Permanent tissue means it has got its own function. Now it will not change its role. How are the cells? So with respect to cells, we can say all the cells are elongated. They have blunt end. Blunt end means the one which is not sharp. Blunt end. But when we look at the TS, they will appear spherical. TS means what? Transverse section. So if I draw this cells, so these are all elongated one. These are all elongated one. But if I cut and I see from the top, top means from the above, if I see, so I will see they will appear spherical. That is what they are trying to say here, that the cells are elongated, blunt end, but spherical in TS. Let's talk about the cell wall. In collenchyma, the cell wall is made up of cellulose, most important part. It is also made up of hemicellulose. Cell wall is made up of cellulose, hemicellulose, and even it is made up of pectin. All the collenchyma cells, they are unevenly thickened. Most important for MCQ. It is unevenly thick only at margins. It means it is not thick all over the body, only at the margin it is thick. The collenchyma, it may have a chloroplast. If it has chloroplast, it will do the function of photosynthesis. And most important, it is found below epidermis of dicot stem. So below the epidermis of dicot stem, you are going to get the collenchyma. So let's understand the collenchyma with a diagram. That will be more easy. So what we are going to do, let us draw the diagram for basically collenchyma. So I draw it in this way. So these are all these cells. These cells are elongated. As we said that in elongation, it appears to be blunt. These are all elongated cells. So I take it in this way. These are all elongated cells. Since they all are elongated, it appears to be blunt. But when I take a TS or the transverse section, then it will appear spherical. So the cells are not evenly thickened. So here I have to just cover it up just to show that they are not thick. So cellulose deposition is not seen everywhere. This is very important. At the margin there are more cellulose deposition, but not equally everywhere. Now in this, let us draw a nucleus. We show this as the nucleus. So there is a nucleus present. There will be vacuole present because it's a plant cell, and plant cell must have vacuoles in them. This all becomes the vacuole. Now in this there will be cytoplasm. So this all becomes cell. This is the Sclerenchyma cells in elongated condition. It means you are observing from the side. So when you observe from the side, they will all appear tall with a blunt end. Blunt end means they are not sharp. Like when you buy a new pencil, so the end is not sharp. So that is called as blunt pencil. So when you sharpen it, it will become very sharp. So this is Sclerenchyma. Let's take the TS. The moment you take the TS, you will understand how it appears. So it is, now here you can see there is a TS diagram. TS basically means we are talking about the transverse section. So here we have taken the TS, or you can say transverse section of the Sclerenchyma. So the cells here, they will not be evenly thickened. So somewhere you will find more deposition of cellulose, somewhere you will find less deposition of cellulose. So this is the most important characteristic of any Sclerenchyma. They will have uneven thickness and uneven deposition. So here you can see these are all the uneven one. See, there is no even distribution of the cellulose. Now in this we are going to draw a prominent nucleus. So there is a nucleus present which is very much prominent. This is the nucleus. There will be a vacuole. So I draw this as the vacuole. So when you are observing it from the top view, you will see all this in the Sclerenchyma. And inside there will be a cytoplasm present. So this is how the Sclerenchyma looks. You know how it appears in case of TS as well as in LS. So this is Sclerenchyma. Let's do the labeling of Sclerenchyma. So very first we will put it in this way. This is nothing but the LS. LS is longitudinal section, and this is nothing but the TS, that is transverse section. So very first I can say that there is a cell wall present. Then I can say that there is a nucleus present. Third, I can say there is a vacuole present. Then there is a dense cytoplasm. Let's talk about the functions. So now we are going to talk about the functions of Sclerenchyma. So very first and the most important function that I can suggest is that this Sclerenchyma is responsible for providing tensile strength. So I can say it provides tensile strength. Sclerenchyma provides tensile strength, gives elasticity and support. Gives elasticity and support. The second function of Sclerenchyma, it is responsible for growth and elongation of organ. Responsible for growth and elongation of organs. Then it is responsible for, so we can say it prevents tearing of leaf. No matter how much fast is the wind blowing, the leaves will not tear, it will not break from the plant, it will never go. And some of the Collenchyma, they sometimes show the presence of chloroplast. And since they have chloroplast, they can do the function of photosynthesis. So this is all what you should know about the Collenchyma. And after this, we are going to talk about Sclerenchyma. Next, we have Sclerenchyma. First, let's understand what do you mean by this word Sclerenchyma. So the word here is scleros, and the word scleros means hard. Then we have the word enchyma. Enchyma means to pour. And among all parenchyma, collenchyma and sclerenchyma, remember it is only the sclerenchyma which is dead. The rest all are living. So let's see the characteristics. So what we are going to do, we are going to put here sclerenchyma. To this sclerenchyma, let's do a shade so that we can write the characteristics. So as far as characteristics are concerned, very first, as I told you, it is dead. So I can say that Sclerenchyma is dead, simple, and it is permanent tissue. So we have dead, we have simple, and we have permanent tissue. When we talk about this Sclerenchyma, I can say that this cell is devoid of protoplasm. So there is no protoplasm as such present because it's a dead cell. When we talk about the cells of Sclerenchyma, they all are compactly arranged. Most important, all the cells are compactly arranged. When I say compactly arranged, it simply means that there will be no intercellular space. There is no intercellular space present in the Sclerenchyma. When we talk about cells of Sclerenchyma, they are highly lignified. Highly lignified means what? There is a deposition of lignin. And if there is a lignin deposited in the cell, then it is considered as the death one. But it shows even thickness. So there is evenly thickened, not like Collenchyma, which shows uneven thickness. So here we can say that it shows evenly thickened. And what about this cell wall? So cell wall will be made up of lignin. Why lignin? Because it is a dead cell. Now let's draw the diagram for Sclerenchyma that will help us to understand in a better way. So for this, let us take it this way. So this is the Sclerenchyma cells. I will be drawing only 4 cells of Sclerenchyma. So these are all the cells. So I take it in this way. So this become the cells here. And this, there will be a covering present. Let us take this way. So this is the important part. Now this is the lignin basically. Deposition of lignin results in death of the plant. In any of the plant cell, if you see lignin deposition, just consider it to be dead. Deposition of lignin indicates only one thing, that the plant cells here, they are going to be dead. This is very important part. All the plant cells will be dead. That is the lignin deposition, and here we put some space. So like this is what is Sclerenchyma. Inside, the nothing will be present, no cytoplasm, no nucleus, nothing, because it is a dead cell. And dead cell will be devoid of protoplasm. So there is no protoplasm present as such. So let's do the labeling. So what you can say, this is nothing but lignified cell wall. This is lignified cell wall. When I use this word lignified, it means the one that is made up of lignin. And here we have something called as middle lamella. So we show middle lamella, plus it will have a primary wall. So we have middle lamella, and what we have is the primary wall. There are two things involved. This is nothing but the TS of fiber. Basically in Sclerenchyma you study two things, the fibers and the sclerids. So here we are talking about the TS. Let's look at the LS. When I look at the LS, I can see something like this. When I look at the LS, it is pointed and tapering both. So this is the LS. LS means the longitudinal section and TS means the transverse section. So again, this is lignified cell wall. If you have seen coconut, the dry coconut that we normally break in the temples, so on its body it shows some fiber like stuff, brownish color. So that is also nothing but this Sclerenchyma. This is the lignified cell wall. This is LS of fiber. LS means longitudinal section. So this becomes the LS of fiber. Let's talk about now fibers and then we can discuss about sclerids. So whenever we talk about fibers, see Sclerenchyma include two things, fibers and the sclerids. So when we talk about fibers, I can say these are long, they are narrow, and most important, they all are thick walled. So long, narrow and thick walled. What about the cells? So we can say that the cells are pointed at both ends. So when we talk about this, look at this. This is the tapering or the pointed at the both end. What about the cells? We can say that the cells are lignified. Since there is lignin deposition, I can say it is a dead cell. So when we look at the LS, under LS the cells appear to be elongated. When we look at the TS, that is the transverse section, it appears to be polygonal. So this becomes the fiber. So what is the role, or I can say the functions of fiber? So the most important function of fiber is it provides mechanical strength. So mechanical strength is provided because of this. It is responsible for conduction of water. So this becomes the fibers. Let's talk about the next important part of Sclerenchyma, that is called as Clarids. So we are having the next part of Sclerenchyma, that is called as Clarids. So Clarids also, they have a lignified cell wall. They have a lumen or small space present. So first of all, with respect to Clarids, I can say that they are isodiametric. So as far as diameter is concerned, it is same. Or even I can say it is polyhedral. So we have isodiametric and polyhedral. How are the cells? So we can say they are elongated and they are rod, even I can say bone, or I can say star shaped. So they are the elongated, rod, bone, or star shaped. From where this clarids develop? So we can say it develops from parenchyma. The parenchyma cells only, when they become dead, they will become sclerid. So all these sclerids, they are thick, hard, and lignified. The covering of the mango seed is of sclerid. And what is the other name for sclerids? So remember, sclerids are also called as stone cells. The other name given to sclerids, they are called as stone cells. So where they are found, so we can say they are present on the covering of seed and nuts, whether it is groundnut or anything. What are the function? So again, let's talk about the functions of sclerids. The function of sclerid is it is responsible for mechanical support. So it is going to provide mechanical support, and at the same time they are responsible for resistance in plant. So this all becomes about Sclerenchyma, which is a dead tissue.